TEER is basically measuring your electrical resistance of the cell layers if they are on top of each other, and then measuring their electrical resistance and seeing if they are tight or if they are loose. If they are tight, they are healthy. If they are loose, then there is some problem. So this is like a mini lab on your device. Like you can test, it's non-invasive. So if you put your cells into it, so the drug or the pharmacist or doctors can see in a real time if this drug or this medicine is put to test. Is it reacting good, or is it improving, or is it leaky, or how the cells are reacting? And in future, we can have these as a personalized medicine too, like individual cells of a person having these on a non-invasive device. So it's basically a lab on a chip.
So the idea was to start taking very specific cell types from individual bodies. That's where personalized medicine also comes in. And like reproducing or recapitulating those signatures in a dish. So instead of going to an animal, you do all of it on a dish, which is easier to control, easier to manipulate, easier to get information in and out. You can do a lot more easier with these things on a chip as opposed to in an animal. Very recent announcements by the FDA and the National Institutes of Health have indicated a reduction in animal testing by 2035, if I'm not mistaken, to completely eliminate animal testing as we go screen for different drugs, which means we need a substitute. If you don't want to test in animals because the translation from animal studies to human studies is really poor, you don't want that scenario for any sort of drug development, be it neurological, cardiac, reproductive system, you name it.
Our partners, WPI, whom we worked with, had these designs which were compatible with their machines. So when they approached Dr. Rajaraman that, hey, and Dr. Rajaraman came up with an idea, oh, let's do this fully fabrication, clean room based.
We've been working with World Precision Instruments for a few years now, and we continue to work with them. And they've been able to translate this rather quickly into something which is highly scalable, because all the development and the designs that we did here and all the process testing and things like that we did here was already ready in terms of commercialization. So that translation from doing it on an academic scale, when you're already developing things that can translate easily into the commercial scale, I think is highly valuable for company-based research.
One of the benefits doing research with the UCF was they have a very good facility for clean room, like very good instruments. And so it helped, and also there are staff and people who are very collaborative.
We can define these things almost on a manufacturing scale, which is very unique in academia. In academia, usually you're doing single device. Most of my colleagues doing device work probably are used to doing single devices at a time, characterize it, produce a paper, and move on. But because we work with a company from the get-go, we are actually starting from the beginning, starting to do it on a larger scale. And I think like developing those kinds of manufacturing-ready processes is, I think, one of the big things that we have done.
Every day is different here. Like you have some successes and many failures, and that's how to improve that one thing. Finding solutions is what is motivating for me to come every day.